everybody, and welcome to our December Superfund Research Program seminar. And I want to welcome all of you on site and also those of you who are joining remotely. And I would say for those of you joining remotely, if you are not part of our list served, it will be good, our email list, send them an email to Nancy, because we will be very, very happy to include you so that you can get emails about our regular activities. And we are really delighted I am delighted to introduce our speakers today, and I am going to introduce first for our seminar, Dr. Barry Rosen. We are very fortunate to count with his expertise, his tremendous arsenic expertise for our seminar today. And oh, sorry, I didn't introduce myself for those of you online, because I think everybody here on site knows me. But my name is Ana Navas, and I am the director of the Columbia University Superfund Research Program. So it's my honor to introduce Dr. Barry Rosen. He's a distinguished university professor at the Herbert Wertheim College of Medicine at Florida International University in Miami. He's been there for 10 years, and before that, he was also chair and professor of biochemistry and molecular biology at Wayne State University School of Medicine in Detroit, Michigan. Before that, he was at the University of Maryland. And for his training, he actually trained in Connecticut, and both for his bachelor and his master, and then also for his PhD, actually. And as I said, he has done tremendously important research on arsenic related to arsenic transport, discovering some of the major role of aquaporins in arsenic transport, major breakthrough in understanding arsenic 3MT, the methyl transferase that methylates arsenic. And we are really privileged to have you with us. But also, I would want to add something else. We are very lucky that he joins often for our journal clubs and seminars, and he's been a great contributor to our discussions remotely. So thank you so much that we can get to you today in person. Yes. Yeah. So which one is the one?
To the top.
Second from the top.
Second from the top.
Okay.
Perfect.
Well, it's a pleasure to be here, and I appreciate the invitation. I've been listening to these webinars for a long time, and I think they're a great thing. I learned a lot from them, and I think my lab is probably listening today, so hi, guys. What I'd like to talk to you about today are how organisms, in particular microbes, have adapted to arsenic to be able to use them in a positive way by using them as antibiotics. And I'm going to tell you two stories today about two antibiotics that contain organic arsenic. One of them is old and one of them is new. And I define old as being more than 3 billion years old. And the new one is only about 2.5 billion years old. So that's the new antibiotic. Page down, not working.
Just click on that thing for a second. And now try.
Yeah, there we go. Okay, so arsenic in the environment has a number of cycles, both cycles of inorganic and cycles of organic metabolism. Many of you are probably familiar with the redox cycles that arsenic undergoes between different redox states. And there's also organic cycles that take place in marine environments where arsenic is incorporated into compounds that are sequestered in marine animals for detoxification. But what I'd like to talk to you about today are the organic arsenical cycles that take place in soil and water, and in particular, cycles of methylation and demethylation of inorganic arsenite. So that's these cycles right over here. These are cycles where inorganic arsenic can be methylated and demethylated. In addition, we use, in addition to the natural sources of these compounds, we introduce them as herbicides and pesticides So we use methylated species on golf courses as herbicides. We feed synthetic aromatic compounds to farm animals as ways of increasing growth. So it's the cycles of the biotransformations that take place here that I'd like to talk to you about. I don't have to tell this audience about the problems with arsenic or the sources of arsenic because everybody here is familiar with it, but just to remind you that arsenic is and always has been #1 on the Superfund list, not because it's the most toxic of these metals. It's not as toxic as lead, mercury, or cadmium, but it's the most prevalent. And so it's the one that everybody is exposed to on a daily basis. And everybody has resistance mechanisms from E. coli to humans. Without those resistance mechanisms, you wouldn't be alive today. So that's why arsenic is #1 on the Superfund list. You get it primarily from rice. This is a major source of dietary arsenic, but we also use it as herbicide. So this one, which is just methyl arsenate or MSMA, is a widely used herbicide. These synthetic organic chemicals like roxarsone are used for chickens, poultry, swine, for the treatment of infectious diseases. It holds down infectious diseases and increases the growth yield of these animals. So there's a lot of arsenic introduced in the environment, both naturally and anthropogenically. Now, arsenic, as you know, is associated with various forms of cancer, diabetes, and other diseases, but probably more important and more critical is the effect of arsenic on infants and children, where the developing brain in fetuses and infants are much more sensitive to arsenic than in adults. So you have developmental delays, you have neurological problems that are caused by arsenic. So this is probably the major concern that we should have now about arsenic. But what I'd like to talk to you about today is my hypothesis, which is actually backed up by substantial data these days, that methyl arsenic, methyl arsenite, the methylated form of inorganic arsenite, is a primordial antibiotic. And it's formed by reduction of arsenate, which is very low toxicity, to trivalent arsenic, which has moderate toxicity, but then it gets methylated by the enzyme Ars-M in microbes or AS3MT in animals to the methylated species trivalent, which is very toxic. And this has antibiotic action in microbial systems. Antibiotic was first termed by Selman Waxman to describe any substance produced by a microbe that inhibits the growth of other microbes. And by that definition, methyl arsenite is an antibiotic. The other thing that is a hallmark of antibiotics is antibiotic resistance. And as methyl arsenite acts as an antibiotic on some organisms, other ones develop resistance to it. So I'm going to tell you about the formation of this and about the mechanisms of detoxification of methyl arsenic. So bacteria transform relatively non-toxic arsenite into highly toxic methyl arsenite. That's antibiotic synthesis. It kills other bacteria. That's bacterial warfare. This is what's going on every day in microbial jungles. But other bacteria develop enzymes to detoxify methyl arsenite, and that's antibiotic resistance. One question that's always come up is, what's the target of arsenic? I can't tell you the target of arsenic in humans because it's probably, there's no single target. It's probably a lot of different targets, but Luis Garbinski, graduate student in my laboratory, recently identified a key target in bacteria, which is the synthesis of the cell wall. So cell wall biosynthesis is the target of a lot of antibiotics like penicillin, ampicillin. In this particular case, Luis cloned genes from a prevalent soil bacterium Schwannella putrefaciens and identified a gene called MurA, which is the first committed step in the synthesis of the cell wall. And this enzyme, MurA, is inhibited very strongly by methyl arsenite. So you can see here growth of bacteria with increasing concentrations of methyl arsenite. You can kill cells. When he took the clone genes from the soil bacterium and introduced them into E. coli, they became resistant. So overexpression of this cell wall enzyme provides resistance to methyl arsenite. And if you look at the purified enzyme, it's very sensitive to both methyl arsenite and the synthetic aromatic arsenicals. And that means that this is one of the major drug targets of this antibiotic. So how is methyl arsenite formed? Well, it's produced by the enzyme AS3MT in humans or ARSM in microbes. And we identified the gene in microbes by cloning genes from a Yellowstone thermophilic eukaryotic alga, Cyanidia, which can grow on solid arsenic. So here you have, this is a picture from the organism in Yellowstone National Park. Here you have solid arsenic, which is being deposited from these effluents from geysers and whatever, and they form solid ocrement, arsenic sulfide. Growing on these, you see this green stuff, that's the alga. So this is really resistant to arsenic. And it's resistant to arsenic because it has this gene, ARSM, which methylates the arsenic and provides resistance. And you can see by multiple alignment of algal enzymes with the human and rat that these are very closely related enzymes. So this is a very close ortholog of the human AS3MT, is an excellent model to use for studies of arsenic methylation. This actually is a very ancient enzyme. So we've been looking at the rise, the evolution of the different arsenic resistance genes. And it turns out that ARSM is the most ancient of all of these genes and proteins. This is actually about 3.5 billion years old. It goes back to almost the origin of life. Other genes, and I'll be talking about some of these, evolved later. Some of them only evolved after the great oxygenation event, which is when the atmosphere became oxidizing. And then you evolved enzymes that could deal with arsenic using oxygen. And at this point also, all of the methyl arsenite that was produced by organisms that have ARSM would get oxidized to non-toxic methyl arsenate. So you have, at the origin of life here, the evolution of ARSM, the rise of ARSM, and it would produce methyl arsenite as a primordial antibiotic. But then, when the atmosphere became oxidizing, which is right here, all of a sudden there was a switch from toxicity to resistance, to tolerance to arsenic. So the difference between ARSM and methylation being production of a toxin and detoxification is whether oxygen is present. So 2.5 billion years ago, there was this switch from production of a toxin to detoxification. So you oxidized it to non-toxic methyl arsenate, but then other microbes, members of microbial communities, took advantage of this oxidized species to reduce it back to methyl arsenite. And so, and I'll show you that in a few minutes. So this species, these bacteria now became the predators in these microbial jungles. The gene arose in bacteria, but by lateral gene transfer, it moved into other members of different kingdoms of life. So as recently as less than 1 billion years ago, by lateral gene transfer, it moved into metazoans and us. So this is the origin of AS3MT, which is our liver enzyme that detoxifies arsenic. So let me tell you a little bit about our studies. I'm going to do a diversion into human AS3MT. And I'm going to show you some crystallography, and I realize that enzymology and crystallography is not the major focus of the research of many people here, and so I'd like to tell you why this is important. Crystallography is what's called the aha factor. You probably heard the story of the blind men and the elephant, that they would touch the elephant and say that the trunk is like a snake, or the elephant is like a piece of rope, or the elephant is like a tree, because they couldn't see the elephant. But when they could see the elephant, they would say, aha, that's what an elephant is. Well, crystallography is the same thing, that if you take a picture of the protein by X-ray crystallography, you can now see where the active site is, how the enzyme works, and this is the aha factor. Being able to see the structure tells you, aha, that's how the enzyme works.
We've trained the eyes.
So what do we learn from this? What does enzymology and structural biology tell us about what AS3MT does and why arsenic is so toxic and carcinogenic? Well, in humans, AS3MT is a phase 2 enzyme that's simultaneously protective against acute exposure and activates arsenic into a potential chemical carcinogen and toxic during long-term chronic exposure. So it has these different effects. And you may ask, well, why did we retain this if it's bad for us? Well, we retained it because it gives protection against acute exposure. But the downside is that it turns arsenic into this potential carcinogen. The majority of arsenic, methyl arsenic, after the first methylation, remains bound to the enzyme. And this is what the enzymology tells you, that it remains bound as MAS3 and only slowly is converted into DMAS, the dimethylated species. So it's this pool of intracellular arsenic which we propose is responsible for arsenic toxicity and carcinogenicity, not inorganic arsenic, not the dimethyl species, but the single methyl species. Because dimethyl species doesn't bind to the enzyme. It comes off the enzyme, rapidly leaves the cells, goes out to the bloodstream and urine, and has very little harmful effect because it gets oxidized rapidly. This is why urinary ratios of high ratios of DMA to MAS3 are considered protective. So the faster you can go from the first step to the second step, the better off you are. So why does that happen? And that's where we learn something from looking at the enzyme in detail. So we have this very fast step where arsenic is methylated the first time, and then a very slow step where it's methylated the second time. So both of these are very toxic and carcinogenic in humans, but the difference is that this is retained in cells and this is not. And you learn that by looking at how arsenic is bound and methylated. So this is the crystal structure of the active site of the enzyme, where arsenic is bound to cysteine residues in the protein, and S-adenosylmethionine is bound with the methyl group poised to transfer to the arsenic. And that tells you how the enzyme works. But if you look at the rates of reaction, it tells you something very interesting and very pertinent to why MAS3 is toxic. So here we have methylation. This is now the human enzyme we're using, a purified human enzyme. And we look at the methylation. So here's total arsenic. You see the inorganic arsenic disappears, and at the same time, you see methylation. So you see rapid formation of the methylated species, slow appearance of the dimethylated species, which corresponds to a decrease in the methylated species, which tells you that it's a precursor product relationship, one methylation, then a second methylation. But if you look here, you see something that is very relevant, and that's that the first methylation step from inorganic arsenic to the methylated species is very fast. But the second methylation step is very slow. So the first methylation step is much faster to produce MAS3 than it is to produce DMA. So that means that there must be some slow step between those two methylations. And it's this slow step that creates a pool of toxic MAS3 in cells. If it gets methylated faster, it comes out of the cells and gets excreted. So as long as you keep the methyl arsenite inside of cells, you're at risk. So why does this happen? And that's because there is a rate-limiting step in this reaction mechanism, and this is what the crystal structure shows you. So let's look at an initial step. We have arsenic bound, we have S-adenosylmethionine bound. The arsenic and S-adenosylmethionine are close to each other, and you get transfer of the methyl group to the arsenic. That's here. But this creates a problem because here you have the S-adenosyl homocysteine, which has to come off the enzyme, and a new molecule of SAM has to bind here, but it can't because this methyl group is in the way. So as long as this methyl group is facing the SAM binding site, you cannot get a second round of methylation. So what happens is that this methyl group reorients the face solvent. And this is a very slow step. So it's this reorientation of that methyl group, which is the rate-limiting step in this reaction mechanism. And it's because of this that methyl arsenite is toxic inside of cells. So it's really methyl arsenite. I'll get rid of this. Can they see this?
I don't think, no, they don't see that.
They don't see that, okay. I see things on the screen that you can't. So anyhow, going back to this, in humans, AS3MT is a phase 2 enzyme. That means that it's simultaneously protective and produces toxic species. The majority of the arsenic remains bound to the enzyme as methyl arsenite and only slowly is methylated a second time. Even though both of these are very toxic, it's that block between the first and second step, which means that methyl arsenite builds up inside of cells, and it's this intracellular pool which is responsible for arsenic toxicity and carcinogenicity. DMA, on the other hand, rapidly dissociates from the enzyme, leaves the cells, goes to the bloodstream, goes to urine, and the faster you can do this, the higher the ratio of the dimethylated to the methylated species is, and that's protected. So the faster you do it, the better off you are. So if you have AS3MT polymorphisms that make the enzyme slower, like these, so here we have 8 different polymorphisms that we've studied that all make the enzyme slower, and they put you more at risk for arsenic-related diseases. So it's really the rate of the enzyme. If we could increase the rate of reaction of this enzyme, arsenic would have very little effect in the body. All right, let me now come back to bacteria and talk about resistance to methyl arsenite. So Paul Ehrlich, who developed the first arsenic organoarsenical drug for the treatment of syphilis, Salvarsan, said in his Nobel Prize address that resistance follows the drug like a familiar shadow. Whenever you develop a drug, you get resistance to it. And that's what's happened with methyl arsenite. Some members of microbial communities produce this toxin in response. Other bacteria have developed resistance mechanism. And the first one I'll talk about is this enzyme Ars-I, which detoxifies methyl arsenite by breaking the carbon arsenic bond and converting methyl arsenite back into inorganic arsenic. This is not so old. This enzyme is only about 2 billion years old because it had to wait for oxygen to appear in the atmosphere because this enzyme uses oxygen. This is the work of Masafumi Yoshinaga, who's a major figure in our group and has done many of the studies that I'll be talking about now, and is also looking for a job if you happen to have one. So he identified from golf course soil, where the golf course was being treated with the MSMA herbicide, methyl arsenate. He identified microbes that could degrade methyl arsenate. And he found that in microbial communities, there are some bacteria which can reduce methyl arsenate to methyl arsenite, and other bacteria that could take that trivalent methyl arsenite and break it down into inorganic arsenic. And this enzyme is catalyzed by an enzyme called Rs, which is a carbon arsenic bond lyase. So here's an experiment that demonstrates that, where bacteria such as Burkholderia, Pseudomonas, Cynorhizobium in the microbial communities can reduce methyl arsenate to methyl arsenite. So if you have methyl arsenite, this is ICP-MS, HPL separation of the species, and measurement of arsenic by ICP-MS. So if you look at methyl arsenate treated with Burkholderia, they can reduce it to methyl arsenite, but they can't take it any further. Streptomyces can break the carbon arsenic bond, but it can't reduce it. So only when you have a microbial community where these things grow together, you go from methyl arsenite to inorganic arsenic. So Rsi is an iron-dependent dioxygenase. We've solved the crystal structure. This is the work of Venkatesh Nader in my laboratory. And the crystal structure here is the AHA factor that tells us how this enzyme works, because here you have iron bound to the enzyme. And iron is bound to 3 critical conserved residues, which are shown here, here, and here in alignment with several of these. So that's the iron binding site. And then you have a flexible loop over here, which moves in turn from solvent to the active site and has 2 cysteine residues, which bind methyl arsenite and move it from solvent into the active site of this enzyme to allow it to be cleaved. So that's the mechanism by which Rsi works. The second resistance that we identified is Rs. We're running out of letters of the alphabet, by the way. So again, you have methyl arsenite being produced and mechanisms of detoxification. In this case, Rs oxidizes methyl arsenite back to methyl arsenate. So you're basically turning it from a high toxic compound into a low toxic compound. And we've solved the crystal structure of that enzyme as well, but I'm not going to go into details of that. Like Rsi, Rs is a relatively young enzyme, only about 1.5 billion years old. Again, because it requires oxygen to do the work, and that's the study of Jen Chen, who's a faculty member in our group, who showed the reaction mechanism is like a mixed function oxidase to take trivalent organoarsenicals, including methyl arsenite, and compounds like roxarsone, which is the synthetic organic arsenical, or phenylarsenite, and it oxidizes them using NADPH and molecular oxygen to form methyl arsenate and NADP.
So Dr. Rosen?
Yes.
Which one will have higher affinity between the H and the I?
Yes, they're both in terms of resistance. They're both about equally effective. The thing is that you develop multiple mechanisms of resistance, and then you start putting them together in single organisms, and you amplify these resistance. So you can get higher levels of resistance by adding these things together. So here you see in cells that don't have the gene for Rsh, you see cells are very sensitive to increasing concentrations of phenylarsenite, but if you put the gene in there, they're now resistant. So this is clearly giving very strong resistance to organoarsenicals. And then the third mechanism of resistance that we identified is RSP, which is a permease that pumps methyl arsenite out of cells. So this is a very simple way to get resistance. You just take the compound, you put it out of the cell. You don't modify the compound at all, so you're not really changing it, and other cells are still sensitive to it, but you're resistant to it. This again is more recent than Rs. but it doesn't require molecular oxygen, so it seems to have evolved during the period before the Great Oxidation Event, although it's still newer than RSM. It's maybe 500 million years younger than RSM. So this again is the work of Jen Shen, who showed that RSP is a methyl arsenite efflux protein. So RSP can pump out methyl arsenite. There are transporters for inorganic arsenic like ACR3 and RSB, and they're not reciprocal pumps. So if you look at cells that are expressing only RSP, they're resistant to methyl arsenite. But cells that are expressing the inorganic arsenic transporters are sensitive to methyl arsenite. And the reverse is true. If they're exposed to inorganic arsenic, RSP does nothing, but ACR3 and RSB give good resistance. So somehow or other, these transport systems can tell the difference between two compounds, inorganic arsenic and methyl arsenic, which are isosteric. They look the same. The only difference is you have a carbon here and an oxygen there. So you have a microbial community here where you have antibiotic production, antibiotic resistance, cycling of arsenic between methylated and inorganic arsenic, and these are emergent properties of microbial communities, where some organisms produce methyl arsenite, some reduce methyl arsenite, some detoxify methyl arsenite. So you have methyl arsenite as an antibiotic, you have detoxification by demethylation, oxidation, or efflux, and this is an emergent property of these microbial communities. In other words, the whole is greater than the sum of the parts. You can't predict how these things are going to work unless you put them all together. I'd like to now turn to the second part of my talk, and this is a new antibiotic, which is newer than methyl arsenite by 1 billion to 2 billion years. And the reason we know that is because it contains oxidized arsenic, which could not have existed before the Great Oxidation Event. So this is a compound called arsenithricin, which is a new arsenic-based antibiotic. And you can see here that it is very similar to glutamic acid. It's an arsenic mimetic of glutamic acid. So glutamic acid has a carboxyl, gamma carboxyl group. Arsenithricin has an arsenic here in place of the carbon. So otherwise, they're very similar to each other. This arsenithricin was discovered by our collaborator, Satoru Ishikawa in Tsukuba, Japan, where he identified this compound as something being made by rice rhizosphere bacteria, Burkholderia, which produces this compound. So as cells grow, they take inorganic arsenic and they convert it into first a compound which has inorganic arsenate in place of the carbon carboxylate of glutamic acid, and then this hydroxyl gets converted to a methyl group with time to form arsenithricin. So Ishikawa identified the compound but didn't know what it was good for. And we had ideas on what it would be good for, and that is that AST, arsenithricin, is a potent antibiotic. So you can see here that it inhibits the growth of E. coli as well as methyl arsenite. And this is rather striking because it's the only pentavalent organic arsenical, or the only pentavalent arsenical, which has high toxicity. In general, pentavalent arsenicals are rather non-toxic. This one's very toxic. It's as toxic as methyl arsenite, which is very toxic. Why is that? Because it looks like glutamate, and it inhibits this enzyme, glutamine synthetase, which is the enzyme for incorporation of nitrogen into glutamine and formation of a intermediate, which is a phosphorylated intermediate, which looks very similar to the arsenithricin. And so arsenothricin binds to this enzyme and inhibits it and prevents formation of glutamine, which is required by most organisms. So it's really not acting by a mechanism like other arsenicals. It's acting as a glutamate analog. And there are other glutamate analogs that do very similar things. For example, phosphoenothiacin, also called glufosinate, which is a commercial herbicide, very effective commercial herbicide. This has a carbon-phosphorus bond instead of the carbon-carbon bond here or the carbon-arsenic bond here. And then there's methionine sulfoxamine, which has a carbon-sulfur bond. These are all very toxic compounds, all inhibitors of glutamine synthetase, and also antibiotics that are formed naturally by soil bacteria. But they aren't particularly good antibiotics. These aren't good antibiotics. This is a much better antibiotic. But this one seems to be much more toxic to plants, which is why it's a good herbicide. So here we see that arsenothricin is a broad spectrum antibiotic. That is, it's effective against both gram-positive bacteria and gram-negative bacteria. And it's also at least 20-fold more effective than phosphoenothiacin, which is that phosphonate analog. So here you see the effect of 25 micromolar arsenothricin compared to 400 micromolar phosphoenothiacin. And it takes more phosphoenothiacin to give less resistance. So you have bacteria, gram-negative bacteria, which are sensitive to arsenothricin. You have gram-positive bacteria, which are sensitive to arsenothricin. We found only 2 exceptions to this. One is the Burkholderia producer of arsenothricin, because most bacteria that produce antibiotics are resistant to their own products. We don't know how it's resistant, but it needs to be, or it couldn't produce the antibiotic. The other one that we found is a Pseudomonas species, which is resistant to arsenothricin. And as I'll show you in a few minutes, that's because it has a resistant gene. And that's very important for the ability to use arsenothricin as a clinical antibiotic. So arsenothricin is effective against pathogens. It's effective against some of the major World Health Organization critical pathogens. One of them is Mycobacterium, and you can see that arsenothricin inhibits the growth of this relative of MTB, Mycobacterium bovis, and is very effective, much more effective than phosphoenothiacin. And we believe that it's also going to be very effective against MTB, but we don't have a BSL-3 facility where we can test it, Mycobacterium tuberculosis. So we're using this as our model. And it does cause tuberculosis in humans by zoonotic transfer from animals, but it's also the major form of tuberculosis in cattle and deer and other farm animals. So it's a very important pathogen. Arsenothricin also inhibits carbapenem-resistant Enterobacter, which is one of the WHO critical priority pathogens. You can see here that, CRE is inhibited by AST, growth is inhibited by AST, whereas phosphoenothiacin and methionine sulfoxamide are not effective against this critical pathogen. So this is really an antibiotic with potential against critical drug-resistant pathogens. On the other hand, it does not appear to be cytotoxic to humans, at least in culture of human monocytes. So you can see that inorganic arsenic is very toxic to monocytes, whereas AST is not. But as Paul Ehrlich said, resistance follows the drug like a familiar shadow. And here we see that Pseudomonas putida is resistant to inorganic, to arsenothricin. I showed you that on a previous slide. If we knock out the gene for ARSN, which is what the resistance is called, from Pseudomonas, they become sensitive. And if we put the Arsen gene on a plasmid, Pseudomonas becomes resistant again, and E. coli becomes resistant. So this is a resistance mechanism for arsenothricin, and it's very widespread in bacteria. You see this Arsen gene in many bacteria. So if you have widespread resistance to an antibiotic, it implies that the antibiotic is also widespread. So we wanted to understand how Arsen works and what we could do to reverse resistance by Arsen. So Arsen turns out to be an N-acetyltransferase. That is, it acetylates the amino group of arsenothricin. And by acetylating it, it no longer can fit into the active site of glutamine synthetase, and therefore it inactivates the compound. It uses acetyl-CoA as the acetyl donor, and we crystallize the enzyme with bound acetyl-CoA and arsenothricin in order to understand how this enzyme works. We can use that crystal structure now to look for things which inhibit the enzyme. And that's what we did in order to increase its effectiveness as an antibiotic, because resistance in pathogenic bacteria is a global challenge. But if we use inhibitors to Arsen in combination with arsenothricin, we can prolong the life of the antibiotic. And this is a commonly used strategy by drug companies to increase the life of antibiotics, inhibit the resistance mechanisms. So we screened in silico, that is, you can take the crystal structure and bind compounds in silico to look for things which can bind to the compound. So we did that with a database of more than 200 million compounds and found some that bind to the active site, at least by computer prediction. Then we bought several of these and showed that they actually inhibit the enzyme. So they inhibit the enzyme non-competitively, and we're trying to crystallize Arsen now with this enzyme, with these inhibitors. But we also found that phosphonates, which are similar to AST, also can reverse resistance. So here's one which is 3-phosphonoalanine. It's a phosphonate which is similar to AST, has an arsenic carbon bond here, phosphorus carbon bond here, and this compound is not toxic to E. coli. So if you have E. coli that's expressing the resistance mechanism, they're resistant to AST. They also are not killed by this compound, AP3. But if you add these together, now AST is killing the cells again. So this compound can reverse resistance to AST. And that's the kind of thing that we're trying to develop. This will allow us now to develop AST as a clinically useful antibiotic. Here you see the crystal structure with AP3, and it binds to the same place that AST binds. So that's why crystallography is very useful. So to summarize this, resistance to available antibiotics against major pathogens is a global challenge. Soil bacteria produce this organoarsenical natural product, arsenothricin, that is a broad-spectrum antibiotic that's effective against major pathogens and even drug-resistant pathogens. The mechanism of action of AST is as an amino acid analog, not as a toxic trivalent arsenical. So its mechanism of action is completely different than other arsenicals. And we believe that carbon arsenic bond compounds such as this may be a new and effective class of drugs, just as phosphonates are one of the major classes of antibiotics and drugs. We believe that arsenates, similar to this, can be the founding members of a new class of drugs. And with that, I'd just like to thank the people who did the work, and they're all watching right now, so I have to be very careful. This is us in Miami. You could be here now, swimming in a swimming pool. And with that, I'd just like to thank you very much.
much. You open for questions, and I know we have one online, so maybe we should, Nancy, if you have read it.
One online from Mirik Stiblo. It says, in vitro cultured cells from some tissues accumulate dimethyl arsenic to a greater extent than inorganic or monomethyl arsenic. Dimethyl arsenic represents a big fraction of total arsenic in some tissues, lung, bladder, of mice exposed to arsenic. Would this contradict the idea that dimethyl arsenic quickly leaves cells' tissues?
Well, Eric, I'm not familiar with that work, so I can't really comment too much on it. I don't know whether we're talking about intracellular trivalent dimethyl arsenite or dimethyl arsenate, the oxidized species, or whether it's inside of cells or outside of cells. It can be in tissues, but not in the cells.
Trivalent, all of them.
Trivalent, all of them, inside of cells.
You can unmute yourself if you want. Yes, yes.
Mirak wants to ask directly. You're welcome.
Yes, inside the cells.
Okay. Well, then we're totally wrong.
Question, you said that its mechanism of action is being a glutamate kinetic, and so it inhibits glutamine synthesis.
Yes.
Would you expect it to inhibit glutamate synthetase as well?
Well,
You could try to repeat that so that.
okay. So the question is, are there, we've shown that arsinothresin inhibits glutamine synthetase because it acts like the substrate glutamate. So the question, I think, is, will other enzymes that also use glutamate as the substrate be inhibited by arsinothresin? And we think that that's possible, but we haven't looked at any others, and we'd like to.
So that I'm particularly interested in an enzyme that adds glutamate to folate metabolites in cells in order to retain folate in the cells. And if it did, then it would totally disrupt folate metabolism.
Well, I think that's a very good suggestion to try. Now, I didn't mention this, but there are also other N-acetyl transferases and Rs operons that look sort of like Rs-N, but aren't Rs-N. And what that says to us is that there are other antibiotics that contain inorganic, organic arsenicals, maybe other amino acid analogs. And so maybe there are some that inhibit other enzymes like that. We're searching for them now. But I think it's a good idea to try other enzymes that use glutamate.
Can I ask one other question? All of these R's alphabet genes, are there any human homologs to any of them?
Well, of course, AS3MT is a homologue of Rs-M, but Rs-M is 2.5 billion years older than AS3MT. But as far as we know, there aren't a lot of human orthologs of these genes. What you might find is some of these genes in your microbiome. And so they may have effects on humans even if we don't have the genes themselves. And then other eukaryotes have some of these genes, just not humans. So fungi have a lot of these genes. Plants have some of these genes, but humans don't. Yes.
Barry, so it's tour de force, and thank you. I have a question and a comment. You mentioned the fast first methylation, and then the second slow, and that the methyl group, once bound to the enzyme, has to
Reorient.
How do you know that?
By looking at different crystal structures. So crystallography is like one frame of a movie. So you have this picture and it says, well, maybe something's going on here. So what you do is you get other crystal structures in other bound forms, and then you start putting them together like frames of a movie, and you can see things moving. Of course, we're putting them together arbitrarily. We don't actually know which is first and which is second, but we put them together in what looks like a logical order, and that's what we did there.
And just a comment. So I've taught pharmacology to first-year medical students for years. I bring with me, I open the course and I bring with me the first edition of Goodman and Gilman, 1940, the very first edition. And I say to the class, if you were here in the class of 1942, let's see what the antibiotics were. Can you imagine that the antibiotics were arsenical? And everybody giggles. But we're back, aren't we?
Right. Well, we're running out of antibiotics.
I know.
So new ones are constantly needed and urgently needed. And if these are a new class, you know, that'd be wonderful.
Oh, please, no, please, you go first.
So the arsenic tea is a natural occurring antibiotics.
Yes.
So compared to the more synthetic form, such as the fourth generation ciprofloxacin, those are currently very important in clinic. So how effective are these AST compared to currently available drugs on the market already?
That's a good question. It requires some studies that we haven't done yet. You need to test them in animals, for example, and we've done no animal studies to date. So what we can tell is effectiveness in bacterial cultures, how sensitive they are, but it doesn't really tell you how good they'd be as a human antibiotic. So we need to do additional studies, and we plan to do those if NIH lets us.
You know, most resistance arise because of too many general usage, and therefore it bears this resistance phenotype taken through as a process of mutation. So in this AST, compared to some of these ASR that the strains that you mentioned earlier, because of the cell wall synthesis, what times of transition in between the phases of this bacterium would you expect that is going to occur?
Yeah, well, you know, most of these resistances were there naturally long ago. You know, they evolved along with the antibiotics. So it's the constant use of the antibiotics that cause amplification of these in animals and humans. But the resistances were there first. That's not to say that new resistances don't evolve, because they do. So there are some of the newer antibiotics that we use evaded resistance for a long time and had to use mutation to produce resistances. So both of those can occur, and they occur all the time. As long as we keep using antibiotics, they're going to occur. So we can stop using the antibiotics, or we can develop new antibiotics, or we can develop inhibitors of the resistances. These are all things that we can do.
Thank you.
I'll try to be quick with my question. Thank you so much. So with the fast methylation and the slow methylation for the first step. Sorry, the
Fast first
and
step, slow second step.
So do you have an crystallographic analysis for instance, arsenic TMD for mice, or compared to humans? And does that support that in mice it's faster both steps? I was wondering, I was thinking, or maybe it's the second step that must be different speeds between humans and animals, because we have more MMA and less DMA as compared to mice. So the question would be, the second step, this rate of the second step, what we think is slow, might be even slower in humans as compared to other animals. And can you see that in your crystallography analysis?
Yeah, well, you know, we're using purified enzymes, so it's difficult for us to compare mice and rats and humans. We can compare their enzymes, but that may not tell you what's happening inside of their cells.
Okay.
So that requires collaboration of people like us with people like you. Or people like Merrick Stiblow.
Yeah, I'm right.
Hi. So you showed the nice pictures of the MMA crystallography, and then you just talked about the DMA. Do you also have the picture, the cartoon picture of the DMA? I didn't see it in your presentation.
And the answer is no, and I'll tell you why. Let me get back to
You could just X that.
Yeah.
Okay. Okay, so,
It's coming.
It's coming.
okay. So here we look at the active site of the enzyme. And what you see is that arsenic and methyl arsenic are bound to two cysteine residues. And trivalent arsenicals bind to cysteines very strongly. But they usually require two cysteines in order to bind strongly. So inorganic arsenic binds to actually three cysteines. It binds to this one as well initially before that disulfide bond is formed. And so it's bound very tightly to the active site. When you methylate it the first time, form methyl arsenic. So here you have methyl arsenic bound to those same two cysteine residues, and it's bound very strongly. Why? Because it's got two coordinations available. But now when you methylate it a second time, you only have one coordination available. And so it can't bind strongly to proteins because there's only one cysteine that it can bind to, and it requires two to form a strong binding site. So DMA comes right off the enzyme. Doesn't bind to the enzyme. That's why we can't get a crystal structure of it.
Okay, thank you so much for the great talk. Now we can relax a little bit, chat around, and then we'll be back at 4 or 5.